Thank you. And I forgot my disclosure slide, but I have nothing to disclose, unfortunately. So Tom already gave the title, and so I'm going to be talking about preclinical versus clinical development of novel therapies for melanoma, using dasatinib as an example of the paradigm of what we oncologists deal with on a day-to-day basis. So the rationale, I can't see the slide here. I have a problem. Yeah, because the computer's not working. Is there... I'm not sure how to fix this. Well, if I move over here...
There you go. That's it.
All right. So the problem in a nutshell is that the incidence of melanoma is increasing, as is the incidence in metastatic disease and death from this disease. Now, there are subsets of patients who do very well with metastatic disease who respond to high-dose interleukin-2 and drugs like ipilimumab for a prolonged period of time. However, most other therapies are associated with a less durable response, and not all of these drugs are very well tolerated. Therefore, we definitely need new well-tolerated drugs for this disease. There are a large number of new therapies that are being developed by pharmaceutical companies, and the question is, how do we choose which ones to start studying, and how do we prioritize our clinical trials? Should all drugs undergo thorough preclinical testing for every disease prior to administering them to patients? Or should we start by offering these experimental therapies to patients who really cannot afford to wait years for their preclinical testing to be done? And what actually constitutes reasonable preclinical testing? How do we get this funded in the current environment? So I'm going to talk about a phase 2 trial that we did with the Src kinase inhibitor dasatinib in advanced melanoma. The rationale for targeting Src kinases at the time, we wrote this trial in 2006, was primarily based on data that we retrieved from the literature. So there are a number of studies that provide evidence that Src kinases are important in melanoma. One of them was published in Oncogene in 2002. Another study, for example, was published by Ruth Halaban here at Yale in 1997, in which she showed that Src kinases are important intermediaries for FGF receptor 1 signaling in association with melanoma proliferation and tumorigenesis. So dasatinib is a multi-targeted tyrosine kinase inhibitor. The list of the targets are listed over here. And this is the IC50 for target inhibition in vitro studies. And you can see that for most of the targets, the IC50 is in the low nanomolar range. For example, CSARC, the IC50 is a half a nanomolar. c-KIT is fairly effectively inhibited, and we'll be talking about c-KIT a fair bit. Dasatinib was given to patients in phase 1 clinical trials, including trials for solid tumors. The mean peak concentration were half an hour to 3 hours after the dose was administered, and the serum concentrations were just over 100 nanomolars when the drug was given at the full dose of 100 BID. The mean terminal half-life is in the order of a few hours. The drug is orally bioavailable, and then it's metabolized by the liver, and the metabolites are inactive. So the drug does have extensive extravascular distribution. Therefore, the serum concentrations might not reflect the intracellular concentrations. Now, in phase 1 studies, they showed that dasatinib inhibited phosphor-Src at very low concentrations in the serum of about 18 nanomolars. We looked at expression of the various dasatinib targets in our melanoma tissue microarrays using a method that David's going to be talking about called AQUA. The y-axis is the expression level on all the different tumors, and the x-axis is just the designation of melanoma versus Nevi. And you can see, based on these means plots, that the expression of CSARC in melanoma was significantly higher than in Nevi. I don't have the p-value up there, but it was less than 0.001, and the same was true for yes. The table at the bottom shows the association with survival on primary melanomas, and we found that both CSARC and yes were associated with poor survival in these patients, providing additional support for trying this drug in melanoma. So our rationale, in summary, was that the phase 1 solid tumor study had a single melanoma patient who had prolonged stable disease lasting 16 cycles or 16 months. There was preclinical evidence, as I said, that Src kinases are important in melanoma. The drug was reasonably well tolerated in the phase 1 trials. The FDA approved this drug for CML, I think, in 2005, and then in 2006, CTEP sent out mass solicitations for phase 2 studies for solid tumors, including melanoma, and we were selected as the site for melanoma. I just want to add, everybody always thinks that our cancer center doesn't do the best job at opening the trials in a timely fashion, but we were by far the first site to open. And dasatinib was made available to us for lab use through a material transfer agreement with Bristol Myers Squibb in late 2008. So it did take a while for us to be able to do any type of lab work. No. Okay. So the objectives of our clinical trial were firstly to determine the overall response rate of dasatinib in patients with unresectable melanoma, to determine the progression-free survival, and to determine expression of potential targets of dasatinib in pre-treatment biopsy specimens, and to look at the association with response to therapy. Eligibility criteria for the trial are listed here, and I'm not going to go through them one by one, but just to highlight the important ones, patients had to have either stage 3 unresectable or stage 4 unresectable melanoma. Active brain mets were not allowed, and this was a first-line therapy trial. Patients could not have had prior chemotherapy, but they could have had up to 2 prior cytotoxic therapies. The trial design was fairly straightforward. It was a Simon's Optimum 2-stage design. In the first stage, we accrued 18 patients, and we determined that if 3 of the first 18 patients achieved either a partial response or a complete response or stable disease lasting more than 6 months, the study would continue to the second stage of accrual. And we decided to include the stable disease of over 6 months because a lot of these targeted therapies really induce what we call stable disease by RECIST criteria. So if a patient has 28% tumor reduction, is that really any different than 30% tumor reduction? We feel it's not, and so we decided for the purpose of this trial to include prolonged stable disease. One cycle was defined as 28 days, and staging studies were done every 2 cycles. We allowed some dose modification. We started off with 100 milligrams BID as the initial dose and allowed 2 dose reductions for toxicity. After the first 17 patients were enrolled, we found that the drug at this dose was too toxic, and we had to amend the protocol starting at lower doses and allowing additional dose modifications. The enrollment demographics of the patients are listed over here. We had 39 patients total, 36 were evaluable. The other 3 had issues either with compliance or comorbidities and really couldn't be assessed. So all of the percentages and analyses are based on this number of 36. 26 patients were enrolled here at Yale and 10 were enrolled at the University of Minnesota. The demographics were fairly classic for metastatic melanoma, predominance of males. We selected patients with a good performance status. Most of them had not had prior treatments. At some point over the course of this clinical trial, it became apparent that c-KIT could be an addictive oncogene in melanoma, so we started enriching the trial for patients with either acral lentiginous or mucosal melanomas because these are the subtypes that tend to have c-KIT mutations. And the staging distribution, again, was fairly typical for what we see. The first 17 patients were enrolled at the full dose, 100 BID. The median time on this dose was only 4 weeks, and we had to hold them in the majority of cases because of toxicity. Primarily, we saw dyspnea, pleural effusions, fatigue, and diarrhea. So we then amended the protocol and started at 70 BID. We were able to keep patients on for longer. Less toxicity was seen, but it was pretty much the same toxicities. This table over here summarizes the adverse events. Most of them were grades 1 and 2, but grade 1 or 2 fatigue for a patient who's on a study for a year or 2 can really be somewhat disabling. There were no grade 5 adverse events, and the most common toxicities, again, were the fatigue, the dyspnea, primarily related to the pleural effusions. In terms of activity, of the 36 patients that we saw 2 bona fide partial responses. One lasted 6 cycles, one lasted 16 cycles. We saw tumor regression that did not quite constitute a PR in 3 patients, one lasting 16 cycles, one lasting 34 cycles. So this patient was on for 2 years and 8 months, which for our disease is a long time, and the other one lasting 7 cycles. There was one patient at University of Minnesota who withdrew after 4 cycles, but he had 27% tumor reduction, and he was continuing to respond. He just didn't want to continue. And then there was another patient who had very transient regression after 2 cycles and then progressed after the 4th cycle. This table over here summarizes the data on those individuals. I'm just going to go through it really quickly. The 2 PRs had quite significant tumor reduction. There were another 2 patients who had close to 30% reduction, which is the RECIST criterion for a partial response. This is the patient who had 9% reduction but was on for a very long period of time. The second column over here is the c-KIT mutational analyses, which Jeff Sklar did for us. Only one patient, the first patient, had an acral lentiginous melanoma and had an activating mutation in exon 13 of c-KIT. This is the CAT scan of that woman. She was a 77-year-old woman with an acral lentiginous melanoma. She had a really nice response in her pulmonary metastasis. This was actually a relatively small pleural effusion, but she was very symptomatic from it. And in fact, in the 6th month on therapy, she was off drug more than she was on drug, and not surprisingly, she progressed. We then treated her with Temodar and sorafenib. Sorafenib is also a c-KIT inhibitor, and she responded to that as well. She's been on it now for approximately 2 years. This is an 82-year-old gentleman who didn't quite meet PR criteria but was on drug for 16 cycles. He had a very large pleural effusion but actually was barely symptomatic. And this is the same man's CAT scan of the neck, where you see this tumor that really never changed in size. It was 24 millimeters for the whole time he was on study. That was about 16 months. But the lymph node clearly became necrotic. And we saw a fair bit of this on the dasatinib patients. Progression-free survival and overall survival are summarized in this slide over here. The median progression-free survival was only 8 weeks, so most of the patients came off relatively quickly. Overall survival was fairly typical for melanoma. The median was 55 weeks, and the mean is actually probably over 67 weeks because there still are some patients in this cohort that are alive, as you can see over there by the tail of the curve. c-KIT staining on these pretreatment specimens. The patients who had tumor reduction, as I said before, only one patient had a c-KIT mutation. That was the one with the acral lentiginous melanoma. All of the others were wild type for c-KIT. And of the patients that had acral lentiginous melanomas, we had tissue in 3 of the 5 patients. 2 had c-KIT mutations. One of them was this patient, and she responded, but the other one did not respond at all. The third one just had expression by immunohistochemistry, but no mutation. Ocular melanomas all had, of the three patients that had available tissue, they all had expression by immunohistochemistry. None of them responded. The 2 mucosal melanoma patients who had tissue for evaluation did not have c-KIT mutations, and neither responded. So there's no clear association between c-KIT mutation and response. Perhaps in this particular patient up here, the one with acral lentiginous melanoma, the c-KIT was driving her tumor, but all the other 5 patients who had some tumor reduction clearly did not have any abnormalities in their c-KIT. So at that point, we decided to go back to the lab to try to figure out exactly what it is that's causing some of these patients to do better than others. And by then, 2 papers had been published on the activity of dasatinib in melanoma in vitro. One study published in 2008 showed that dasatinib in vitro studies blocks migration and invasion of melanoma cell lines. It inhibits Src kinases at very low concentrations, but there was little to no effect on proliferation and viability. They did their viability studies looking at exposure for 72 hours. There was another paper that came out of Ireland where they exposed their cells for 5 days. And there they found that in one of the 5 cell lines, the IC50 for dasatinib was 35 nanomolars. The other ones were all over 1 micromolar. So we tried to do that again in our own hands. We took 8 melanoma cell lines, and we only exposed them for 72 hours, but you can see that there are clear differences between 2 of the cell lines, UROB and ULAC, where the IC50s are in the 100 to 300 nanomolar range. All of the others, the IC50s, are well over 4 micromolars. The drug was penetrating the cell, and this is evidenced by these studies where you see downregulation of 2 of the targets, the phospho-PDGF receptor beta and phospho-C-Src, both in the sensitive cell line, UROB, and in a more resistant cell line, USOC. We looked at synergism with chemotherapy, and I think Ruth came up with that name. So we looked at synergism with chemotherapies. We looked at cisplatin, which is the most commonly used chemotherapy for melanoma in Europe, and dacarbazine, which is the most commonly used drug in the United States. And when we added chemotherapy to dasatinib in the resistant cell lines, it didn't do anything at all. But when we added it to in the more sensitive cell lines, you could see that, in fact, there was some sensitization to dasatinib, and we saw synergism for only the 2 sensitive cell lines, UROB and ULAC, more for cisplatin than dacarbazine. The bar graphs over here just simply show the same thing. The combination index of less than 1 is considered synergism, and you can see over here that either drug alone was less potent in terms of decreasing viability than the combinations. That's just different concentrations. So we sent our cell lines down to MD Anderson for them to profile them with reverse phase protein arrays. And I'm going to take a minute to talk about the arrays with the hope that maybe we can pressure our Keck facility to start using this technology over here, because I found it to be very useful. So these arrays are simply glass slides coated with nitrocellulose, and you send the lysates down there. There's a robot that embeds the lysates onto the slide with serial dilution so that you can get more quantitative levels. You can put up to 1,020 different lysates on a slide, and of course every slide has protein controls that have been established by ELISAs and other methods, and you get computerized output for every single sample, and everything is compared to the standardized curve for that particular sample for that particular protein. You can look at total protein, phosphoproteins, and so on. You can only do one protein per slide. So when I sent the lysates down there, at the time they were doing just over 100 proteins, and they took our 8 lysates that we sent them and profiled them for their 100 and something proteins. The protein that was most differentially expressed between the sensitive and resistant cell lines was caveolin-1. So this is the caveolin-1 expression. We did 3 biological replicates, the caveolin expression in ULAC and UROB, the 2 more sensitive cell lines. You really don't see it at all in the more resistant cell lines, and that was confirmed by Western blot. So we then went back to our patient specimens and collected what we could. We used David's aqua method to study the levels of caveolin-1 using this quantitative immunofluorescence. And you can see this is an example of stronger caveolin-1 staining. This is weaker caveolin expression. This is an attempt to do a chi-square, and I say an attempt not because I don't know what button to push on the statistical program, but really the numbers are very small. So of the 6 patients that had some tumor regression, 5 had high caveolin expression, and of course you saw less caveolin expression on the patients who had progressive disease. Again, I want to caution everyone, the numbers are very small, and I'm not sure what conclusions we can make from this. But in the meanwhile, 2 other studies were published showing an association between dasatinib sensitivity and caveolin expression in other diseases. This first paper is breast cancer cell lines, and the second one is on ovarian cancer cell lines. All of this is in vitro work. So in conclusion, single-agent dasatinib has modest activity in unresectable melanoma. Our study did not meet the predefined criteria for clinical activity as written into the protocol. Toxicities were common but manageable. Responses were seen in 1 of 2 acral indigenous melanomas. Acral indigenous melanomas that had c-KIT mutations. The other patients that had tumor reduction and tissue accessible for c-KIT did not have mutations. There is an ongoing clinical trial in patients with acral indigenous melanoma being conducted by ECOG. The study does involve tissue collection, and assessment of c-KIT mutational status will be done on those patients. The cell line IC50 is higher than the concentrations that are probably achieved in vivo. And I say that a little tongue-in-cheek because you do have the extracellular distribution or the extravascular distribution of the drug. But ideally, we should have done all of these preclinical studies prior to initiation of the phase 2 clinical trial. We would have had a better idea of what we were dealing with. It's not clear if SOC was adequately inhibited in these patients. It probably was based on the phase 1 clinical trials, but it's also unclear if SOC inhibition itself is enough to mediate anti-tumor effect with this drug. The association between caveolin-1 expression and sensitivity should be restudied in additional patient cohorts. As I said, we are collecting specimens from the ECOG study, and we'll be able to assess caveolin expression on those. There's also a phase 2 study combining dacarbazine and dasatinib being done at UCSF, and hopefully we'll be able to get specimens from that trial as well. We need to do additional studies to determine if caveolin-1 is merely a marker of sensitivity or if it's a mediator of sensitivity. Future combination studies should be based on both activity and lower concentrations, perhaps enough just to inhibit SOC, but not enough to induce all this toxicity that we saw. And we should also do that on molecularly defined subsets of patients. I want to point out that even though the results here weren't too impressive, the studies with the EGFR inhibitors in lung cancer, for example, really only showed response rates of 10% or so. But when you select the patients properly, and enrich for patients with EGFR mutations, you really do see a higher response rate. So perhaps we're dealing with something similar over here. So combinations with chemotherapy specifically might allow us to use lower and more tolerable doses of dasatinib. I've already said that one. And then I think we also need to address the difficulties in obtaining drugs for preclinical testing. We couldn't get the drug quicker from Bristol-Myers Squibb. Part of it was due to the time that it takes just to figure out an MTA between Yale and Bristol-Myers. But certainly, if this drug was available to us for some lab studies, we might have been able to do a better job of selecting the patients appropriately. So I'd like to thank all of the people who participated in this work, primarily Mario, who helped with the trial design and supported the study all the way through, David for the correlative studies, Ruth for all sorts of intellectual input and cell lines, etc. Annette was the statistician. Jeff Sklar did the c-KIT mutational analyses. All the members of the clinical research team are listed over here. John Rhee referred us patients for this trial. University of Minnesota was excellent with accruing patients. CTEP sponsored the trial. Funding for the preclinical work came from the Milstein-Meyer funds. Bristol-Myers was also very helpful, and of course the Yale Spore and Skin Cancer helped with the tumors. Lucia did all of the lab work, or most of the lab work, and other members of my lab helped as well. MD Anderson did the reverse phase arrays. Be happy to take questions.